Every single day, your body is doing something pretty much miraculous. We're talking trillions of cells dividing, making perfect copies of themselves, just to keep you going. It's this incredibly precise, controlled process. It's almost always flawless. But what happens when that perfect biological machine makes a mistake? That's what we're going to get into today. How one tiny error in just a single gene can kick off a truly catastrophic chain reaction.
So this is really the big question, isn't it? How does one single cell going rogue manage to create something as complex and devastating as a tumor? To really get a handle on the disease, we've got to first understand the beautiful, intricate process that it messes with. So let's start with how the system is supposed to work. Okay, so think of the cell cycle like a super sophisticated self-driving car. It's got a destination programmed right in: create 2 healthy new cells. And under normal conditions, it just works. It's perfectly engineered. It knows exactly when to go, when to slow down, and when to stop, all because of this incredibly complex internal program. Right, and this car isn't just driving blind. Not at all. It is loaded with multiple redundant safety systems. We call these the cell cycle checkpoints. They're like these little surveillance stations that are constantly monitoring the whole process. They're checking the engine, the fuel, the road ahead, you name it, all to make absolutely sure every single step of the journey is done flawlessly before it even thinks about moving on to the next.
So what's actually in the driver's seat here? Well, it really just boils down to 2 fundamental opposing systems that are encoded in our genes. And you can think of them, pretty simply, as the accelerator and the brakes. These 2 systems are always working together in this delicate balance to control the pace of all cell growth.
And here's where that car analogy really clicks into place. On one side, you've got proto-oncogenes. These are your gas pedal. They're the ones giving the go signal, pushing the cell to grow and divide. And on the other side, you've got tumor suppressor genes. Those are the brakes. Their whole job is to say stop and bring the whole cycle to a halt whenever it's needed.
Let's take a closer look at that gas pedal. Now, proto-oncogenes are totally normal. In fact, they're essential genes. They're what we call the positive regulators. They create the proteins that send the signals saying, hey, it's time to move forward, time to copy the DNA, time to divide. Honestly, without them, we couldn't grow or heal. They are literally the engine of progress.
And then, of course, you have the brakes. Tumor suppressor genes are the negative regulators. You can think of them as the cell's safety inspectors. The proteins they make can slam the brakes on the cycle at those critical checkpoints. This gives the cell time to make repairs, or, if things are really bad, to take itself out of the game entirely. They are the guardians of stability. So you have this beautifully balanced system, right? A system of go and stop. But this is precisely where things can go so horribly wrong. This is the moment when that finely tuned machine starts to sputter and malfunction, when the wiring for the gas pedal and the brakes just starts to fray. And this is the key. The whole thing starts with a mistake, a single change in the DNA sequence, a mutation, and that one little error leads to a faulty protein. This one malformed part, whether it belongs to the accelerator or the brake system, is enough to throw the entire machine completely out of balance. And one small error isn't just an isolated incident. It actually creates an environment where, as the researchers put it, subsequent mistakes can occur more readily. It's a snowball effect. One tiny mistake gets missed, gets left uncorrected, and that paves the way for a complete catastrophe down the line. To really see how this snowball effect happens, let's zoom in on one absolutely key player, a protein called P53. It comes from a tumor suppressor gene, and it's so important that it's often called the guardian of the genome. In our car analogy, P53 is the master inspector at the most critical checkpoint, making sure every car that passes is totally roadworthy. A healthy, functioning P53 has 3 incredibly important jobs. First, it is constantly scanning the cell's DNA, looking for damage. If it finds an error, bam, it hits the brakes and calls in a repair crew of enzymes. But, and this is crucial, if the DNA is too damaged to be fixed, P53 makes the ultimate call. It triggers what's called programmed cell death, or apoptosis. It sacrifices that one broken cell to save the entire organism. So it's the ultimate failsafe, right? A perfect guardian. But what happens when that guardian itself is damaged? What happens when the master inspector is asleep at the wheel? This number, it's just staggering. A mutated, non-functional version of the P53 gene has been found in more than half of all human tumors.
Think about that. This isn't just one potential problem among many. It is arguably the central point of failure in the story of cancer.
And the consequence is immediate and devastating. Without a working P53, the most important safety checkpoint, the G1 checkpoint, is completely compromised. The brakes are gone. So the cell, no matter how damaged its DNA is, just sails right through the stop sign and barrels ahead into division, taking all of its mistakes with it.
And now, that car is truly out of control. The accelerator is basically stuck to the floor, the brakes are shot, and the inspector is nowhere to be found. We are now on the inevitable path from a single broken cell to a full-blown tumor. So here's how that final crash happens. First, the checkpoints fail. Then, those damaged cells, the ones that should have been stopped, they start dividing like crazy, passing on their mutations and even picking up new ones along the way. These mutated cells reproduce way faster than their healthy neighbors, crowding them out until they eventually form a mass of non-functional rogue cells. And that's a tumor.
And that right there, that is the answer to our question from the beginning. From lung cancer to skin cancer to brain cancer, this is the common thread that ties them all together. A complete breakdown of the cell cycle's controls, which leads to the one thing they were designed to prevent, uncontrolled cell growth. The machine has literally turned against itself.
Which leaves us with a final and pretty profound question. When you think about it, we're not fighting some foreign invader like a virus or bacteria. We're fighting our own cells, our own fundamental biological processes that have just lost their way. Understanding this internal failure is for sure the first step. But wow, does it reveal the true scale of the challenge ahead?